Hi everyone. I hope you've been enjoying the first day of MDF's virtual conference. I'm Tanya Stevenson, the CEO at MDF, and I'd like to welcome you to the very special time in our program when our industry partners come together to share about their myotonic dystrophy programs and their cutting-edge work towards treatment and a cure. This is a chance for us to hear directly from them about their approach, their process, and their progress. Today we're going to hear from Amo Pharma, Harmony Biosciences, Avidity Biosciences, and Dyne Therapeutics. In this virtual format, we've asked our speakers to record their presentations in advance, so unfortunately we won't have the opportunity for questions right now. However, all of our industry partners, as well as many of our sponsors, do have booths, and they will be available to answer your questions live this evening from 4 to 5 p.m. And if you can't get there tonight, tomorrow morning, 8 a.m. to 9 a.m., 12 to 1 p.m., and at 5 to 6 p.m. Pacific. They will be live in their booths to chat with you. You can find their booths on the Exhibitors tab of the conference website. We hope you enjoy the presentations, and we look forward to seeing you later in the conference. Take care.
Hello everybody, and welcome to this presentation by Amo Pharma at the 2021 MDF Virtual Conference. My name is Ibs Mahmood and I'm the CEO of Amo Pharma. Amo Pharma is a UK-US company researching a novel investigational medicine called AMO02 as a potential therapy for the congenital onset form of myotonic dystrophy type 1. We were introduced to the potential for AMO02 in congenital DM1 by Professor Luba Timchenko at Cincinnati Children's Hospital in 2015 and conducted an initial phase 2 clinical study in adolescents and adults with congenital DM1 in 2017 and 18. We are presently conducting a potentially pivotal study in children with congenital DM1, and we will update on that later in this presentation. As you may know, DM1 is caused by a CUG RNA repeat that is present in cells around the human body in individuals affected by congenital DM1. This repeat RNA interacts with a number of protein enzymes within cells, one of which is called GSK3-beta. GSK3-beta is an enzyme that controls the formation of tissues. When the DM1 repeat RNA joins with this enzyme, the activity of the enzyme increases, and increased GSK3-beta enzyme activity causes the generation and regeneration of body tissues to stall. So how does AMO2 work? Well, AMO2 is a traditional medicinal drug compound that is taken once a day as a strawberry-flavored drink. When AMO2 is taken by mouth, it travels in the bloodstream to all parts of the body, including the brain, muscles, the guts, heart, and so on. Once AMO2 arrives at the tissue, it disrupts the CUG repeat RNA and excess GSK3-beta enzyme. In this way, AMO2 can potentially remove the cause of congenital DM1 in all tissues of the body. This slide summarizes the effect of 12 weeks of treatment with AMO02 in 16 adolescents and adults with congenital and childhood onset DM1. We used a variety of ways of measuring the effects of AMO2. Measurements conducted by the clinician running the study, the caregivers for the participants, or tests of muscle function. The results showed that compared to a period of placebo treatment at the start of the study, AMO2 improved symptoms according to all the key measurements taken. This is some more detail of the change in symptoms shown during 12 weeks of treatment with either a low or a high dose of AMO02. AMO02 appeared to produce an improvement in most symptoms of congenital DM1, This broad and rapid onset benefit was accompanied by relatively few adverse events. Amo Pharma would like to thank regulatory agencies in multiple countries for giving permission to study AMO02 in congenital DM1. The FDA has been very helpful in the US, and we were very pleased to see FDA granted fast-track status to congenital DM1. The UK regulators have also recognised the serious need in congenital DM1 by designating AMO02 as participating in a new innovative licensing and access programme. Amo Pharma would also like to thank investigator sites and families affected by congenital DM1 for their incredible hard work in running the present double-blind placebo-controlled study of AMO02 in children with congenital DM1 during the current pandemic. This study started in the first part of 2021 and is now around 50% enrolled, with sites across the world involved or joining soon. As this study completes in 2022, participants are entitled to transition to an extension study in which all participants will be given AMO02 for 1 year. If you would like more information on Amo Pharma, you can go to the contact page on our website at www.amo-pharma.com. If you would like more information on the current pivotal study of AMO02, you can go to a dedicated website to that study at www.reachcdm.com. That's REACHCDM.com. You can also meet us at our virtual booth at this meeting. Thank you so much for listening, and thanks to MDF for allowing our participation.
Good afternoon, everyone. On behalf of the team at Harmony Biosciences, I'd like to thank the Myotonic Dystrophy Foundation for the opportunity to present at their annual conference. My name is Jeffrey Dano, and I am the Chief Medical Officer at Harmony Biosciences, and I am joined today by my colleague, Bill Jacobson, Senior Director of Clinical Development. Harmony is a pharmaceutical company. We are headquartered outside of Philadelphia, Pennsylvania, and we're dedicated to developing and commercializing innovative therapies for patients living with rare neurological disorders who have unmet medical needs. We were established in 2017, and we became a public company last August and are now a fully integrated company with about 160 employees. Most importantly, patients are at the heart of everything we do at Harmony. We've been listening to the myotonic dystrophy community very closely, and over the years, through multiple patient-focused meetings and one that Harmony held this year, a patient advisory board, we see that the key themes of the non-muscular symptoms of excessive daytime sleepiness, or EDS, fatigue, and cognition resonate very much with the DM community, and they're prevalent symptoms with high impact on daily functioning. A word about histamine and sleep-wake regulation. There are chemicals in the brain called neurotransmitters that send signals between nerve cells called neurons. One important one is histamine. It's made in a small area deep in the brain called the hypothalamus. And histamine is a major wake-promoting neurotransmitter. It also affects the release and increases the release of other neurotransmitters in the brain that have effect on wakefulness, as well as other key functions such as attention and cognitive function. Pitolisant is a first-in-class medicine with a novel mechanism of action. It works by increasing histamine levels in the brain. Histamine is not only a major wake-promoting neurotransmitter, but it helps to stabilize states of sleep and wakefulness. Pitolisant was discovered and developed by our partner company, Bioprojet, and then was approved in Europe as Wakix in 2016 for the treatment of narcolepsy in adults with or without cataplexy. Then Harmony took the file and submitted to FDA, and it was approved in the US in August 2019 for the treatment of EDS in adults with narcolepsy, followed by the approval of the cataplexy indication last October. This represented the first and only product approved by the FDA for patients with narcolepsy that is not scheduled as a controlled substance by the DEA. WakeX is an oral tablet. It is taken once daily in the morning upon awakening. With that, I'll now turn the presentation over to Bill Jacobson to provide highlights of our phase 2 clinical trial in patients with type 1 myotonic dystrophy. Bill?
Thank you very much, Jeff. It's my pleasure to present to you our phase 2 clinical trial. I'm going to talk about our primary objective, which in this study is to evaluate the safety and efficacy of pitolisant compared to placebo in treating excessive daytime sleepiness, or EDS, in patients with type 1 myotonic dystrophy. Our primary endpoint in this study is going to be the change in the average sleep latency, which is the amount of time it takes one to fall asleep, based on a special sleep test called the Maintenance of Wakefulness Test, or the MWT. And we'll be looking at comparing this from the beginning of the study until the end of the study and seeing if there's any change that will be due to pitolisant, which will be compared to placebo. The MWT is a special type of sleep study that's performed in a sleep laboratory to measure how long one can actually stay awake. We have a number of secondary endpoints that we're looking at. Myotonic dystrophy is a multisystemic disorder that impacts many things other than the muscles. We're going to be looking at and evaluating fatigue, cognition, or brain fog. We're going to be asking our clinicians, our investigators, for their assessment of the severity of sleepiness over the course of the study. And very importantly, we're going to be asking patients to do the same thing. How sleepy do they feel at the beginning of the study and at the end after having been exposed to the investigational product? We'll also be studying the overall burden of disease. Now, there are a number of inclusion criteria to ensure that we are studying the correct population and to ensure that we're studying a homogeneous population. All of this information will be available at our virtual booth if you're interested in looking in more detail. I just want to highlight a couple here. We're going to be studying adults with genetic confirmation of myotonic dystrophy and doing a couple of assessments to ensure that they actually are suffering from excessive daytime sleepiness. Importantly, one of the criteria that we want is the ability to walk independently with or without an assistive device. So we're excluding patients who are wheelchair bound, but certainly anyone using a cane, a walker, or a brace would certainly be able to participate. There are also a number of exclusion criteria that we have to include. There are many things that can contribute to excessive daytime sleepiness, and we want to ensure that the EDS that we're seeing is actually due to the myotonic dystrophy itself. So one of the things that contribute to EDS is inadequate sleep. So we're asking patients to keep a sleep diary in the early part of the study, in the screening part, to ensure that they're getting adequate amounts of sleep and their EDS is not being caused by a lack of sleep. Another very important issue are sleep-related breathing disorders, such as obstructive sleep apnea. Now, many patients with DM suffer from these disorders, and we're not excluding them, but we are going to ensure that the sleep-related breathing disorder, if present, is being appropriately managed. Drugs that would interfere with the action of pitolisant and some other things are listed here. And again, you can look at these in detail. We're happy to discuss them with you if you'll visit us at our booth. There is one key area I do want to discuss because in our discussions with patients, this has become an area of obvious concern. Many patients with myotonic dystrophy type 1 suffer from some kind of cardiovascular or heart abnormality. Many of them have implanted heart devices, and there seems to have been an impression that many of them have been excluded from previous studies simply by virtue of the fact that they have an implanted device. We are actually not excluding implanted devices unless they're there to treat serious heart disease. Devices that are there either to monitor the heart or that are there to prevent heart problems would certainly be allowed. Safety is a very important issue for us, so we will be monitoring cardiovascular safety during screening and throughout the study. There will be repeated electrocardiograms. and we will ask you to wear a Holter monitor, which is essentially a electrocardiogram that you wear while you're doing your daily activities, and allows us to record you for about 24 hours. A number of standard procedures that we go through in all clinical trials: informed consent, medical history, physical exam, and so on. But there are certain things that we're doing to ensure that you meet the criteria that we need for this study. So I've already mentioned the need to maintain a sleep diary. We will be doing genetic tests for DM1. I've just discussed our cardiac monitoring. There will be cognitive testing, a number of questionnaires, and we will ask people to provide evidence that any sleep-related breathing disorder is being appropriately managed. I'm sure many of you are interested in where the sites are for this study, and this map shows you that we're distributed across the country. We have a number of sites in the US. Two of them are actually open and actively recruiting. One is in Atlanta, and the other is in Indianapolis, and we continue to initiate sites in real time. For those of you who are located, who are not local to one of the sites, I want to assure you that we will provide travel expenses to the site that is most convenient to your location, and take care of travel and accommodation with our concierge service. We're also looking at opening sites in Canada, and we expect that to take place sometime probably the end of this year, or more likely the beginning of next year of 2022. And we are actively working to initiate that study in Canada as we speak now. At the end of the study, which is a double-blind study where neither the investigators nor the subjects know whether they're taking active drug or placebo, patients will have the opportunity to enter into an open-label long-term extension study, where they will all be receiving appropriate doses of pitolisant, and we will be collecting additional data. So that will be available to all subjects who complete the study and are eligible for entry. And for those of you who'd like to learn more about the phase 2 trial in myotonic dystrophy, I would encourage you to visit Harmony at the exhibitor showcase at this virtual meeting. We have a number of resources available to you, including this infographic, the slides that have been shown in this presentation, and we'd be happy to answer questions if you have any concerning the study that we're undertaking. If you are unable to visit us at the booth, at our virtual booth, I would encourage you to email us at clinicaltrials@harmonybiosciences.com. We'd be happy to answer any questions that you have about the study, sites, participation, and so on. Once again, I'd like to take the opportunity to thank the Foundation for the opportunity provided to us to present our study to you, and I look forward to answering any questions you might have or providing you with additional information. Thank you.
Hi, my name is Lisa Ackerman, and I'm the Neuromuscular Therapeutic Area Head at Avidity Biosciences. I want to thank the MDF for the opportunity to present today, and I'm excited to share some of the information with you about Avidity, our science, and our work with DM1. So this is just to say that I'll be making some forward-looking statements during the presentation. Avidity is a biotech company that is based in La Jolla, California. Our mission is to develop innovative new drugs to improve the lives of individuals impacted by rare and serious diseases like DM1. We view the drug development process as a partnership with patients and communities we serve, and we look to partner early and collaborate often during every step of the process. Our trials and programs are designed with inputs from patients and families so that we can make sure that we are meeting their needs and expectations, and that we are generating the best evidence to show that our therapeutics have a meaningful impact in patients' lives. So today I'm going to talk about our drug that we are developing to treat DM1. As you know, DM1 is caused by a mutation in a gene called DMPK. The mutation causes hundreds or thousands of nucleotide repeats to build up at the end of the DMPK gene. When the mutant DMPK gene is transcribed into RNA, the RNA also contains these repeats. The repeats are toxic to the cell and impair normal cellular function. The ultimate result of this toxicity are DM1 disease signs and symptoms that can affect multiple organs in the body. A substantial amount of these disease signs and symptoms are in the muscle tissue, including skeletal, smooth muscle, and heart muscle, as shown in this slide. These include myotonia, muscle weakness, pain, GI issues, abnormal heart rhythm or function, and respiratory or breathing problems, resulting in a significant decline in quality of life. This is an important point because our drug was developed for DM1 specifically to go to the heart and muscle tissue and to reduce the amount of toxic DMPK RNA in these cells. We hope that by reducing the toxic DMPK RNA levels, that there will be a decrease in some of these signs and symptoms. The drug that we are developing to do this is called AOC1001. AOC stands for Antibody Oligonucleotide Conjugate, and it was designed to go to the muscle and heart tissue and decrease the amount of toxic DMPK RNA. So AOC1001 is actually made of two building blocks that are shown in this slide, an antibody and an oligo, and they each have very different functions or roles. The function of the antibody is simply to deliver the oligo to the muscle cells. So you can think of it as a bus that is carrying its passenger, the oligo, to muscle and heart cells. As you know, antibodies are naturally occurring proteins made by the body to fight infections, but they are also commonly used as drugs because they can be made to target and bind to a specific protein in the body. They are also very well characterized for safety and are the foundation for over 100 approved drugs, many of which are for chronic conditions. So the role of the oligo part of our drug is to target the toxic DMPK RNA and reduce its level in the cell. Now, oligos are short strands of DNA or RNA, and they can be powerful drugs because they can be made to specifically bind to a disease-causing RNA and change its expression or how the body is making the protein. There are currently multiple oligos that are approved as drugs, and many of them are disease-modifying therapeutics. For AOC1001, the type of oligo that we're using is called small interfering RNA, or siRNA. It works by harnessing a natural biological process in the cell called RNA interference. RNA interference is a mechanism that is used by your body to fight off things like RNA-containing viruses. What happens is the siRNA is loaded into a protein complex, shown here as a Pac-Man. Once loaded in the protein complex, it will chew up its target RNA. So in the case of AOC1001, it chews up the mutant DMPK RNA. So this slide sort of puts this all together and shows how AOC1001 works. The antibody we are using sticks to a certain receptor that normally sits on the outside of a muscle cell. This receptor is called the transferrin receptor. When the antibody finds to and binds to the transferrin receptor, the receptor pulls it into the cell. Once inside the cell, the oligo is let go and it can do its job, which is bind to that protein complex and chew up the DMPK RNA. So I'm just going to show two slides of some data from experiments that we have done in the lab with cells or animal models. This slide shows the results of a study that was done in monkeys. The monkeys were given a single IV infusion of AOC1001, and we looked to see what happened to the levels or the amounts of DMPK RNA in the thigh muscle and the calf muscle over time. Each time point we looked at is shown at the bottom of this graph, and time zeros before any drug was given. So you can see on this graph that at 4 weeks after a single IV infusion of AOC1001, the amount of DMPK RNA measured had decreased by about 75%, and it stayed down for at least 12 weeks after this single dose. So based on this animal data, we are encouraged that the effect of AOC1001 may last at least 3 months, which would mean that in the clinic it could be given in about 3-month intervals. So this slide is also a study that was done in monkey. Here we looked at different types of muscle tissues, including skeletal muscle, diaphragm, and the heart. In all of these different muscles, we saw about a 75 to 80% reduction of DMPK RNA. So our animal studies showed that the drug was working the way we had intended it to work, and that these data supported moving this program forward into clinical trials. So I'm excited to be able to share with you today information about our upcoming clinical trial that we have called MARINA. It is a phase 1-2 trial that will enroll adults with DM1, and it will be started later this year. The 3 main goals for the MARINA study are to evaluate the safety and tolerability of AOC1001, to understand the pharmacokinetics, which basically means how the body is processing and breaking down AOC 1001, and to evaluate the ability of AOC 1001 to reduce DMPK RNA in muscle biopsies. So this is going to be a placebo-controlled trial, which means some participants will receive active drug and others will receive placebo. The active drug and placebo are both given by IV infusions. The participants, doctors, and study staff will not know who is receiving active drug and who is receiving placebo. So there are two parts, Part A and Part B. In Part A, participants will be given a single dose of either AOC 1001 or placebo and will be followed for 6 months. After the 6-month follow-up period, they can decide to enter an extension trial where everyone receives the active drug. In Part B, there will be 3 different dose levels, and participants will receive either AOC 1001 or placebo 3 times. After the treatment and follow-up periods, they also can decide if they wish to enter the extension trial and receive active drug. So the trial is going to enroll both males and females between the ages of 18 and 65, and all participants must have a confirmed genetic diagnosis of DM1. This study is being run at multiple sites across the U.S., and they're being selected based on their experience with caring and managing for people with DM1. So for further details about the eligibility criteria or our sites, please visit clinicaltrials.gov. So Marina was designed in partnership with DM1 patients and families to ensure that we were meeting their needs and that we were minimizing barriers to clinical trial participation. As a result of direct input from you, we have put in a number of different elements in our clinical trial. These include the use of home health care and telemedicine. So what this means is that some of the visits, participants will have the option to have a nurse come to their house to complete the visit or have a telemedicine visit. We are also using a travel concierge, and this service will help arrange and prepay for some of the airfare, lodging, and transportation. Participants may also be reimbursed for reasonable trial expenses during the trial. So I'm going to close just with this picture of some of the Avidity team members and their faces of the company that are working to address the unmet needs of patients. We are so grateful for the collaboration with the Myotonic Dystrophy Foundation so that we can better serve this community and bring forth meaningful treatment options. So if you have any questions, please do visit the Avidity booth or drop into the sponsor and exhibitor showcase. And you can always connect with us through patients@aviditybio.com. Thank you so much.
Hi, I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. First, I want to thank the MDF Annual Conference organizers for inviting me to present today. It's a privilege to be presenting here. Today, I'll talk about our FORCE platform and data from our Myotonic Dystrophy Type 1 program. These are our forward-looking statements, so please take a moment to view them. At Dyne, it all starts with our mission. We are laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with DM1 being a priority. Our mission is core to who we are and what we do every day. A commitment to understanding the community of people living with muscle diseases, and DM1 in particular, and ensuring that their perspectives and experiences are included in our drug development programs is central to our work each and every day. We've created a multifaceted program involving community webinars, conference presentations, fireside chats, and other outreach to raise visibility and gather input to advance our drug development efforts. Over the past few years, we've been validating our platform and technology, including our ability to target delivery of potential therapeutics to muscle, and have generated exciting preclinical data across multiple indications and disease models. And our focus today is on sharing our data from our DM1 program. We're building Dyne as the leading muscle disease company to deliver for families living with DM1 and other muscle diseases. Shown here are 4 core strengths to help us deliver on that. First, we intentionally designed our FORCE platform to overcome the current limitations of muscle tissue delivery with modern oligonucleotides. The goal for our DM1 program is to develop a therapy that will lead to a reduction in toxic nuclear DMPK, correct the splicing defects, and enable relatively infrequent dosing to potentially stop or reverse disease progression. Second, our focus is on rare muscle diseases. We have generated very exciting preclinical data that shows the power of our technology across diseases and in DM1 in particular, and I'll highlight some of that data in this presentation. The third is delivering for patients. We will leverage our muscle disease focus to advance our programs as fast as we can to the clinic. We've shared that we plan to submit 3 INDs between the end of this year and the end of next year, with our DM1 program anticipated in the early part of that timeline. And finally, we have exceptional in-house and advisory expertise with deep knowledge of DM1 and novel therapeutics, and a world-class scientific advisory board, which includes luminaries such as Dr. Charles Thornton. On slide 5, we describe our FORCE platform. Our platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment, that we've engineered to bind to a receptor called the transferrin 1 or TFR1 receptor that is found abundantly across the surface of muscle cells. If you look at the Y-shaped structure on the left of this slide, that is how a full antibody or monoclonal antibody looks. Our FORCE platform uses the top piece, the Fab, which drives binding to the TFR1 receptor on muscle cell walls. We believe Fabs have several advantages over full antibodies or mAbs, including smaller size, which helps with distribution to muscle tissue. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our therapeutic oligonucleotide to enter skeletal, cardiac, and smooth muscle to target the genetic cause of disease. At the bottom here, we have a linker. We link our Fab to the potential therapy with this linker. The linker molecule connects the Fab to the antisense oligonucleotide, which gets in the nucleus and again is designed to target the genetic basis of muscle diseases we are trying to treat. In the case of DM1, our potential therapy targets toxic nuclear DMPK RNA, which I'll discuss in more detail in upcoming slides. Historically, the challenge, as stated earlier, has been the inability to get enough of these antisense oligos into the muscle cells. Our FORCE platform is designed to solve this delivery issue and deliver therapeutic payloads into muscle cells by attaching the ASO to our Fab, which enables targeted delivery to muscle. On slide 6, you'll see Dyne's FORCE platform is designed to target the genetic basis of DM1. As you all know, DM1 is an autosomal dominant triplet repeat disease of the DMPK gene. In DM1, the toxic number of repeats form large hairpin loops that remain trapped in the nucleus, forming nuclear foci. The toxic nuclear RNA acts as a sponge, which binds splicing proteins, forming foci and causing abnormal splicing events in many genes, including genes important for muscle health, such as chloride channels, calcium channels, and other muscle structural and functional proteins, such as BIN1. This abnormal splicing across multiple genes leads to the clinical phenotype for DM1. As you see on slide 7, the FORCE platform targets toxic RNA stuck in the nucleus to address the genetic basis of DM1. This is the ideal point for therapeutic intervention. We rationally selected a single-stranded ASO to traffic or get into the nucleus and target toxic nuclear DMPK RNA, reduce nuclear foci, and release splicing proteins, which enables normal mRNA processing and potentially stopping or reversing disease progression. Now, turning to some data utilizing the FORCE platform. We studied our lead candidate in DM1 patient cell lines with approximately 380 and approximately 2,600 repeats, and a patient cell line with about 380 repeats as shown here on slide 8. Dyne's candidate demonstrated robust dose-dependent DMPK knockdown, reduction in nuclear foci as represented by the reduction in red foci in the panel shown here on the right-hand side, And this resulted in correction of splicing defects as measured by BIN1 exon 11 exclusion, which is the histogram in the middle of this slide. On slide 9, in DM1 cells with about 2,600 repeats, which represents profound disease severity, we also saw robust dose-dependent DMPK RNA knockdown, reduction in nuclear foci, and BIN1 splicing correction. We developed an innovative HTFR1 DMS XL mouse model to evaluate pharmacodynamic, or PD, and pharmacokinetic activity, or PK activity. In fact, we believe our work with this model really sets a new standard for evaluating PD in DM1. As you see on slide 10, on the left-hand side of the slide, this model represents a severe DM1 phenotype with more than 1,000 repeats. You'll see that the mice in this model express the human TFR1 and carries a human DMPK gene. On the right-hand side of the slide, you can see how this enables us to evaluate the ability of our lead DM1 candidate to reduce toxic nuclear human DMPK RNA. It also has the potential of increased translatability to patients who accumulate this toxic nuclear DMPK RNA. Essentially, this model helps us understand the potential benefit to treating patients. Multiple genetic studies in the field have indicated that aiming to target 30 to 50% knockdown in toxic DMPK has the potential to be disease modifying. On slide 11, you'll see the data we've reported previously where we evaluated our candidate in this HTFR1 DMS XL model after 2 doses at 10 milligrams per kilogram for each dose, evaluated at day 14. Our candidate demonstrated a significant toxic human nuclear DMPK knockdown, 60% in the heart, 56% in the diaphragm, 54% in the TA, and 39% in the gastroc. Turning to slide 12, the histogram on the left-hand side is the heart KD data, which you saw on the previous slide. And consistent with this, on the right-hand side of the slide, you can see that our candidate demonstrates an approximately 40% reduction in DMPK heart foci at 14 days. We expanded our analysis in the HTFR1 DMS XL model to evaluate treatment with a single low dose of 10 milligrams per kilogram and evaluate at a longer time period, as you see on slide 13. In these data, our candidate demonstrated sustained reduction in DMPK at 4 weeks, 51% of the diaphragm, 46% reduction in both the heart and the TA, and 42% in the gastroc. And in these studies, our candidate was well tolerated. What this shows us is that we see very similar robust knockdown of toxic human nuclear DMPK RNA sustained with twice the duration at half the dose. This reinforces the advantage of the FORCE platform to enable targeted delivery to muscle of oligos with low and infrequent dosing. As we've shown, FORCE is designed to address the genetic basis of DM1 by targeting toxic nuclear DMPK RNA. These data demonstrated that a fully human FORCE conjugate can correct the DM1 phenotype of patient-derived myoblast cultures with a range of repeats, including those representative of severe DM1. It can reduce toxic nuclear human DMPK foci in the cardiac muscle of HTFR1 DMS XL mice. And it leads to sustained knockdown of toxic nuclear human DMPK in this mouse model after just a single dose. We're very pleased that these data strongly support further development of our DM1 therapeutic candidate, including a planned clinical study. So this concludes the presentation. Thank you again to the MDF for the opportunity to present today. And thank you to the patients and families in the patient community that partner with us to advance treatments.